This video I'll talk about different styles of trees that are used in evolutionary biology. You'll see lots of different types of trees, and in a previous video I discussed about how there can be a lot of different ways trees can be drawn that don't matter. Different versions of a tree don't impact its information at all. There are features of trees that do contain information, and different features and different ways of drawing out a tree do impact what we can learn from it and how we understand it. Previous video talked about dendrograms. They're a type of tree. Is not an evolutionary tree. It's just used to indicate similarities. There are many other types of actual evolutionary trees. Again, dendrograms are built using cluster analysis. Darwin drew the first actual evolutionary tree. You should always know that Darwin not only was the first real evolutionary biologist, but drew the first evolutionary tree. In phylogenetic trees, we reserve the term phylogenetic tree for trees that represent evolutionary relationships. Throughout this movie, I will use lots of different terms that are used to describe styles of trees. I'm not so concerned that you know exactly the name for a given style tree. What's most important that you understand how to interpret different styles of trees, and the names and what exactly they're called, people debate or have different conventions. All of these are I'll call all of these phylogenetic trees because they represent evolutionary relationships. Key to defining phylogenetic trees are the establishment of clades, and so phylogenetic trees are often called cladograms. So a cladogram is a dendrogram that represents evolutionary relationships. So this first distinction I'll draw on, a cladogram is a dendrogram. Dendro just means tree, but a cladogram is a type of tree diagram that is particularly focused on evolutionary relationships and groupings of clades. So briefly, you might want to pause the video to review a little bit of phylogenetic tree vocab. It's worthwhile noting that the root of trees can be drawn in different ways, but it's always at the base of the tree. So cladograms are phylogenetic trees that represent only the branching pattern. They represent the clades and the branching pattern. The lengths of the branches do not matter, and the style of the tree doesn't matter in this case. You'll notice that the tips of the trees, the tips of the branches are all at the same level. Typically, a cladogram is going to show extant species, or species that lived at the same time. You could put fossils, extinct species, onto a cladogram, but often we'll use a different style for that. Again, the branches do not have any meaning. The length of the branches cannot be compared or contrasted. You can't compare the length of this branch to the length of that branch and make any inference or understanding from it. All that matters are where the nodes are, and where the clades are, and what is the nesting structure of the clades. And most trees, especially cladograms, will have extant species. Those are species that are currently alive. Unknown species, or rather extinct species, are represented as nodes. They are inferred. We don't actually know when this species is or what it looked like, but we infer that species must have existed sometime in the past. So extant species on the tips of cladograms, extinct species are the nodes. Nodes are used to represent extinct common ancestors. So this node is the extinct common ancestor of chimpanzees and humans. This next node down here is going to be the extinct common ancestor of gorillas, chimpanzees, and humans. It wasn't a gorilla, but it was some sort of primate that then, after divergence, one branch became gorillas, the other became human and chimpanzees. The root is the node at the very bottom, and that's the common ancestor of all the species on the tree. So many trees don't have all of the taxa neatly lined up at the top. Many do have branch lengths that vary, and the variation in the branch lengths is an important feature. Some trees have uneven branch lengths because they include fossils, and when a species is extinct, the branch ends. So here, this is a tree showing horses. Most species of horses are extinct. So here is a branch that ends here when all species in this lineage went extinct. Here is an extinction, 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 extinction. Only up here do we get horses that are the extant horses that are currently alive. Some phylogenetic trees have branches that vary in length, and they do have all the species shown are extant. And in this case, the branches are indicating the amount of difference or change that has occurred between species. For example, these types of phylogenetic trees often use genetic information, and the length of the branches is often proportional to the amount of point mutations that have occurred within DNA sequences used to build the phylogenetic trees. For these types of trees, you always need a scale. So the first tip when you see a tree like this is the branches vary in length. They always have to have a scale which tells you how to interpret differences in the length. So in this case, you need to track down where the scale is, and it is up here. Occasionally, these trees are set up so all the branch lengths are equal. They do some statistics in order to make all the branch lengths equal, and then you still know that something is going on, that the branch lengths are important because there is a scale indicated there. So here's another example here. The scale is down here at the bottom, telling you that the lengths of the branches are important. Chronograms are phylogenetic trees that represent time. So in this case, we have a tree over here, and then a time scale is here. The geological epochs, Jurassic, Cretaceous, are indicated on the scale. So usually when, again, as in previous examples, if time or branch lengths are important, then there has to be some sort of scale bar to indicate it, or axis to indicate what's going on. Often when time is involved, it is on a long geologic scale, and so you're dealing with these long geological time periods going back to when dinosaurs were around. In order to put time onto a tree, you need some sort of way of getting time or dates that require some sort of way of dating fossils or dating rocks. Often that's radiometric dating. Could also be using samples from particular geological strata that can then be referred to something that has been dated with a radioisotope. So in order to put time on a tree like this, you need to have fossils in order to be able to date them. In this case, these nodes are indicating divergence, and there are also some branches that terminate, indicating extinction. These nodes here, you can actually interpret them as they are the inferred time when the lineage is actually split. So it's here, this clade here of mammals is thought to have split from marsupials around 140 million years ago. Let's see, it says 140 there, but we can also extrapolate off to the scale here between 125 and 146, so about 140 million years ago, maybe 130. Chronograms, here's another example of a chronogram. This one is using fossil evidence. The tree itself was built using DNA, so sometimes you can use a combination of fossil evidence and DNA evidence. Most usually we prefer to build trees these days based on DNA evidence or using a lot of DNA evidence, but then we can cross-reference fossils in order to put dates on our trees. The scale down here tells us that we're dealing with a time span. 0 is the present. This is 17.5 million years ago. We can look at the scale and we can see over which time period divergence happened and when divergences happened. So in this clade down here at the bottom, There's a period from a little longer than 10 million years ago to around 7 million years ago, so 4-ish million years. There was no divergence recognized in this clade, or at least there's no evidence of extinct species. We don't know. This is in the past. This is all being inferred. But what looks like that this lineage, there was a single population or a single species that for 4 million years there was no divergence, or nothing died and became fossilized. And that tells us about the other lineages. We then have divergence over the next 5.5 million years ago until the present. Then a lot of divergence happened and all these species in the clade developed. Similarly here, there's a clade here of 3 foxes, and around 2.4 million years ago, this lineage here, this population here, split off, and one branch became the Sechuron fox, and then another branch split into 2 different species here over the course of about 2.4 million years. That divergence happened. One thing that's important to remember is that a lack of divergence does not mean no change. So there's just a single line here. There's no divergence of populations into subspecies or species. It doesn't mean change wasn't occurring, just that there was no splitting of the phylogenetic tree. It's important to remember on phylogenetic trees, tips can represent many different things. In this case, it's not species on the tips of the trees, it's actually orders. So when we do taxonomy, species are what we often talk about in ecology and evolution. Species are organized into genera or the genus. Genera are organized into family. Families are into order. So order is a fairly higher level part of the taxonomy. So each one of these are different orders of mammals with many families, many genera, and many species within them. And these diagrams, they're not that common. They're called spindle diagrams. The width of the bar indicates the number of species that are thought to have existed at any given time. So this is indicating that there are currently a lot of bats, though in the past this spindle is narrower, so it broadened over time. This means that the number of bats within this order increased over time. Over here, Monotremata, those are duck-billed platypus. This line is thin, never gets that wide. This indicates the fact that there have never been that many species of monotremes. Legomorpha, same thing. That's rabbits. Hares, not that many species over time. The line is a consistent length. Rodents are over here. This is fairly wide and gets wider, indicating that in the past, a fair number of species of rodents and increasing number over time. Again, these are called spindle diagrams. Most often show up in paleontology. Here, these spindles stop here. They don't extend to the end. So the short length indicates extinction. Time is indicated here, and there's a scale. So time is indicated here, and then there's a scale to help you interpret the width of the bars. So this is telling us about the amount of biodiversity, or the number of species, the number of taxa within the group, so we can actually estimate based on the width of the spindle how many taxa there are. Species richness is just one way of gauging biodiversity, but it's a very common one in evolutionary biology. And species richness, and it's often discussed how many species there were in a group in any given place or any given time. So this bar here, the width of this bar indicates 50 families. So we can look here, we can compare the length of this bar to our frogs here. The bar looks a little shorter. Looks like there are less than 50 families. In this case, we're not dealing with species richness, we're dealing with family richness. Same idea, species richness is the total number of species. Families is the total number of families in an order. So it looks like there's never been more than 50 families in frogs. Maybe in the past there's a little closer to this. In lizards, currently it looks like there's about 50 families. Sometime in the past there were a few more. Birds, we can fit 2 bars here, almost 3, so there's maybe close to 125 or 150 families. And there were times, though, when there were much fewer families, and that increased relatively recently. So only in the tertiary period, very recently, have we had a large number of birds diverge.